Y'all is getting tough in here. All right, let's see. I don't think anybody's on today. Please, please, please do your attendance. Please do your attendance. We got some people on, okay. So please do your attendance. Do not forget to do your attendance. All right. So, like I was saying earlier, not everybody I think was in here, but I went ahead and uploaded the chapter 7 unlabeled mechanisms for y'all. So the really good thing about these that I uploaded is, if it would load, hopefully it will eventually load, that I put one per page, and the internet is slow today. And what I did was I left the figure that it came from on there so that you can kind of look at it and on like scratch paper, start jotting down what you think is happening. Then you can go to your textbook and go find that figure and check your work, right? So you can see if you're on the right track or not.
Is it the same ones that are in the PowerPoint too?
The same ones in the PowerPoint, same ones in the book,
Okay.
same ones here, same ones that will be on the exam.
Same ones that you need to know.
Yeah.
Remember.
You don't have to memorize it.
I know you have to
What
understand.
you said, you're just gonna put this figure in what you explain.
I could. I could. I might ask you questions about the figure. I might not tell you to explain it to me. I might ask you specific questions about it. Like, what kind of mechanism do we see? Where's the intermediate in the mechanism? All kinds of different things that I could ask. Okay. All right. So, oh, and I forgot a joke last time. So I owe y'all 2 today, right? All right. Why do watermelons like big weddings? Big weddings. Because they can elope. Let's see. What did one eye say to the other eye? Something between us smells.
All right, I like the jokes today.
Thank you. I kind of liked him too. All right, so we went over that. I think we're ready to start, right? Any questions? I feel like there was something else I was supposed to tell y'all, but I'll either figure it out or I won't. Okay, so we talked very, very generally about all kinds of different mechanisms for cell signaling, right? So now from here on out, what we're going to do in each of the sections is we are going to go through specific receptors and mechanisms. Do you have to know the details of each of the major mechanisms? Yes, you do. Yes, you do. You do. So befriend your dog and explain it to your dog. You know, anybody, whoever, doesn't matter. All right, so the first one we're going to talk about are G protein-coupled receptors. So G, I always do that. The first time I draw, I always do that. G PCRs, G protein-coupled receptors. So when you hear somebody say G PCRs, that's what we're talking about. There are a lot of abbreviations in the following sections. There's a table of the abbreviations defined in your textbook. Yeah, you're going to need to use it. Okay, you're going to need to use it. I will try really, really hard to spell out most things. Like, I'm not going to spell out GTP. I'm not going to spell out, trying to think what else. I don't remember, but I'll try to spell out most things, okay? But you do need to get comfortable with knowing what different things are. Okay, so our G protein-coupled receptors typically are found in sensory perception. So right, most of the time you have a ligand that comes in and binds to a receptor and induces a conformational change. That conformational change is propagated through the plasma membrane into the cytoplasmic part of the molecule and induces some kind of downstream effect, right? There is an exception to that in vision, right? What's our ligand for vision? Photons, light. It is light. So it's not a traditional ligand, but it's still a G protein coupled receptor. So in G protein coupled receptors, you have 7 transmembrane alpha helices, and each of the alpha helices is represented as kind of a barrel here. And all G protein coupled receptors basically have their N-terminus that are extracellular, so are sticking out into the extracellular space, and then they basically snake. So do you see how here's the N-terminus, here's our first alpha helice, it would go down, you can't see it, there would be a little loop, and then we come up to our second alpha helice. We have a little loop on the extracellular space, third alpha helice, little loop on the cytosolic side, and then we go up to the fourth, right? And it continues to snake up and down that way until you get to the last one, the 7th one. And then the 7th one, the C-terminal tail is sticking out into the cytosol. So this is why we call them serpentine receptors, because they snake up and down inside of the membrane, right? And the other kind of common feature of all of these G protein coupled receptors are that they are typically glycoproteins. What does glyco refer to? Sugar. So they're proteins that have sugar molecules attached to them. And that's what you're seeing here. A carbohydrate is a sugar, right? Same thing. So these different sugar moieties help to facilitate recognition, right? So you can have a common, a very, very, very highly conserved common domains inside of G protein coupled receptors. And then if you change these loop areas and you change the carbohydrate functional groups, now you can make them very, very specific for different ligands. So you can have very highly conserved parts of the molecule, and then you can have very, very divergent parts of the molecule. So it allows for a common mechanism, but for very high specificity on ligands, okay? All right. So what I want to do is I want to show you, I'm going to start with this, and if we have time, I'd like to end with it as well. But it's very detailed, but this is what we're going to go over today. So we're going to start with just the structure in the animation. And then we're going to go through how basically all G protein receptors signal. You might get a little lost. It's okay. You can always go back to this animation at any time. And we're going to talk about the details in class. But this gives you a really nice big overview. Try not to blast y'all out, but that there's enough volume.
Our perception of vision, smell, and taste are all
Hi.
mediated by G protein coupled receptors, or GPCRs. GPCRs show an evolutionarily conserved structure consisting of a central core made up of 7 transmembrane alpha helices. In the cytosol, GPCRs interact with a membrane-bound protein complex called a heterotrimeric G protein, which...
Okay, so the GPCR that we have talked about so far is only this portion right here. So it's a G protein coupled receptor. It is by definition a what? A receptor, but it is coupled to G proteins. Okay, so be careful that you don't label them correctly. This is the receptor. These are the G proteins. Okay.
Consists of G alpha, G beta, and G gamma subunits. The G alpha subunit is a GTPase and can hydrolyze GTP to GDP. When the GPCR is not bound to the ligand, it is bound to the inactive GDP-bound G alpha beta gamma complex. In most GPCRs, an extracellular signal induces a conformational change in the GPCR protein. The conformational changes in the activated GPCR stimulate the exchange of GDP for GTP in the G alpha subunit. GTP bound G alpha dissociates from the heterotrimeric complex. Both G alpha and G beta gamma can then initiate distinct downstream signaling pathways. In some cells, different GPCR-mediated signals are integrated in response to different extracellular stimuli. Liver cells, for example, must respond to the hormones glucagon and epinephrine in order to regulate glucose levels in the bloodstream. Both glucagon and epinephrine activate a specific G alpha called GS alpha after binding to the glucagon receptor or the beta-2-adrenergic receptor.
Okay, let's stop for just a second. Right, I told you it was detailed. You're gonna have to go back and watch this again. Like, I highly recommend going back and watch this again. So glucagon, when do you produce glucagon? Low blood sugar, right? When you're sleeping or you haven't eaten in a while. How about epinephrine? When do you produce it? Fight or flight, bears coming in the back of the classroom, right? Okay. Both of those two events lead to what overall physiological response? Like, at the very end of this pathway, this crazy pathway that we're going to go through, what do you want to happen? Glucose in the bloodstream. We want to increase glucose in the bloodstream. That's exactly right. So whether you haven't eaten in 8 hours, your blood sugar's going low, your brain needs some food, right? Or the bear just ran into the back of the classroom. You're going to have to run for the next mile to get away from him. Just outrun me. You'll be okay. And then you'll be okay. But in order to run for a mile, what do you need? Glucose. Where are you going to get glucose from? No matter what happened, whether it was glucagon or epinephrine, where are you getting glucose? Not fat storage, but glycogen. Glycogen in your liver, right? That's our immediate, quick. Lipids are for long term. Like, if you go into starvation mode, you will start hydrolyzing lipids, but we're not talking about that. We're talking about quick, a minute later, we can bump up our glucose levels, okay? So what you're gonna see is that even though we have two different hormone ligands, we're gonna get the same downstream effect or in product effect, right? So do we have different receptors? Yes. Now, I'm gonna spoil the surprise. I'm gonna tell you there's a third receptor that also binds to epinephrine, but it's not a beta-2 androgenic receptor. It's an alpha receptor. So it also responds to epinephrine, but it has a different pathway. These two have a shared pathway. How do we know that they have a shared pathway? What are they both bound to? G protein S alpha, right? So if your receptor is bound to the same G protein, it's going to induce the same downstream signaling events, right? And that's the whole point. We want glucagon to induce the same response as epinephrine for glucose metabolism. Not for everything else, right? Because when you're sleeping and you haven't eaten in a while, do you want your heart rate to go up? No. But you do want to metabolize glycogen and get glucose, right? So we have some shared pathways, and then we'll have some non-shared pathways. Okay, are ready to start again? All right.
After exchanging GDP for GTP, GS alpha undergoes a conformational change in a specific region of the protein called the switch 2 helix, so that it loses affinity for G beta gamma and interacts with an enzyme called adenylate cyclase. This interaction.
Do y'all remember adenylate cyclase? What does adenylate cyclase do? Adenyl ATP converts it into the cyclic form. Now cyclic AMP is a second messenger that can go on and induce downstream effects, right? Okay.
It stimulates the adenylate cyclase, resulting in the production of the second messenger molecule, cyclic AMP. At low levels of cyclic AMP, protein kinase A is bound to a regulatory subunit within an inactive tetrameric complex. As cyclic AMP levels rise, Cyclic AMP molecules bind to the regulatory subunits, inducing a conformational change that releases the active protein kinase A monomers. Protein kinase A activation triggers several downstream pathways that all lead to glucose export into the bloodstream. One of the substrates for protein kinase A is the cytoplasmic tail of the beta-2-adrenergic receptor. The phosphorylation of the GPCR by protein kinase A and other regulatory kinases provides a docking site for beta-arrestin, a protein that results in downregulation of GPCR signaling and endocytosis of the receptor.
Okay, let's pause for a second. This is a super complex video. If you got your receptor activated, right, it means that you need glucose. But do you want those receptors to be permanently on? No. So one of the effects of initiating this cascade has to be to come back up and inhibit the cascade, right? So you want to have some downstream effect, but you also want to have feedback inhibition. So that's what we're seeing right here. We're seeing a form of feedback inhibition. It's not the only one, but it is one of them. So how are we feedback inhibiting this process? When we phosphorylate the receptor then associates with another molecule, which induces endocytosis of the receptor. Where's the receptor now? It's in the cell. It's in basically a little lipid bubble. Is it exposed to ligands? Is it going to signal? Nope. It's basically shut off. And that's what you want, to receive the signal, induce the change, shut it off. Okay. There's another pathway that it happens, and we'll go over that. But this is one of them. There's another.
The over 600 GPCRs in our body recognize a wide variety of signals, such as light, compounds we can taste and smell, and hormones, to enable our cells to perceive and react to environmental cues.
Okay. It's a really great video, but there's a lot of detail in it. So we're going to go over it. If we do not have time to watch it again, you need to watch it on your own. And if it makes sense, if the whole thing makes sense to you, you're good. If there are parts of it that don't make sense, go back and study those parts. Okay? All right. Now. Okay. And as you're studying and as you're looking at that video, remember that you can come here and you can go and look at any abbreviations that don't make sense, right? Okay, so the first thing we're going to talk about is this beta-androgenic receptor. So we're going to pick one and we're going to start with that one. So beta-androgenic receptor is a GPCR. So this GPCR, which is a receptor, binds to the ligand epinephrine. When that binding occurs, you're going to induce a conformational change. So if you look, the blue diagram is where the molecule, how the molecule is oriented in the membrane when it has no ligand bound. So it was crystallized without a ligand. That's its conformation. You give it a ligand, it's epinephrine, and what happens? You induce a conformational change. Where do you see some really big changes happening? Look down here. You see some big differences between the blue and the green, right? So that's exactly what we expect to see, some really big changes on the cytosolic side. And so you have some up here where the ligand binds. So you'll have the helix movement there. And you also have movement down here on the cytosolic side. And it's these movements, these initial binding interactions that propagate the conformational change that we see down here on the cytosolic side. So it's absolutely 100% ligand dependent. So you can have something like a natural ligand bind, and you have the natural response to that, right? We can actually capitalize on that in the pharmacological industry, right? We can make receptor agonists and antagonists. A receptor agonist is a mimic that mimics exactly the ligand and induces exactly the same physiological response. And I'm going to show you some examples and why this is really important. An antagonist is the opposite of an agonist. An antagonist binds with high affinity, high specificity, but basically is like a suicide inhibitor. It binds and then nothing happens. You block the receptor from being able to signal. Both things are really important when you're thinking about treating patients, right? Depending on what the receptor is and what you want to do, sometimes you want to artificially stimulate the receptor, and sometimes you want to stop the receptor from signaling at all. So I'm going to give you some examples. So here are our basic molecules that are going to bind to our beta-2 androgenic receptor. So epinephrine, norepinephrine, dopamine, these are all natural physiological receptor molecules, ligands. So now what we want to do is we want to make an agonist. These are molecules that we've created in the lab that will bind to the receptor and induce the same exact conformational change. So these mimic natural ligand, right? So what do they do? Isoperotenol. Anybody know what this is? Albuterol? You know what albuterol is? Anybody? It's a steroid. Where do we find it? Where do we put it? Inhaler. Inhaler. So this is, you know, if you see people who have asthma and they have a little inhaler, so that's what's in their inhaler. Okay, let's think about this. What do you think what does the inhaler do? Most of y'all probably know what an inhaler will do. It'll relax your airway, right? So it prevents airway constriction. So why would you want that to happen if, for example, you were releasing the hormone epinephrine? I'm trying to run from the bear, right? What's going to help me run from the bear? I get more oxygen into my lungs. I get more oxygen into my bloodstream. So what do I want to do? I want to open my airways. So I can make a ligand that looks very similar to epinephrine. How similar is that? Right? And I can mimic that behavior. You can also have the opposite of that. So these are antagonists. Antagonists are going to bind to and prevent the receptor from signaling. So depending on which receptors it targets, you can actually, this one is one that will actually prevent vasoconstriction. Right? So normally you can express this in order to constrict blood vessels in certain parts of the body so that you move blood to your vital organs and things like that when you're under emergency flight or fight. So what I want to do is I want to block that from happening, so that will end up opening my blood vessels and reducing my blood pressure. So this one can reduce blood pressure. But you can make, how many different GPCR receptors do we have from the video? A lot. So you can make ligands, artificial ligands that bind to those specific receptors and do certain things, right? Because epinephrine is going to bind to many different receptors and induce lots of different things, right? Increased blood pressure, increased heart rate, opening up your airways, dilating your eyes, so on and so on and so on and so on, right? So you can target specific ones in order to inhibit or promote certain activities. Okay. All right, so you remember how we kind of talked about in the video that both glucagon and epinephrine did the same thing. And I don't know if I put this in your, do you have this picture in your notes? I didn't remember if I added it before or after I posted your notes.
It's not on that slide.
It's on that slide. Okay. It's in your textbook. It's in your textbook. That's my fault. I can post it if y'all want me to post just this figure. I can do that. Okay, so we have glucagon and we have epinephrine. What are all these things supposed to, in the end, do? Increase our blood sugar, right? That's my shorthand. Increase, maybe I don't want to do that. We'll say blood sugar. How's that? A little bit better about that. What y'all think about this class? All right. So doesn't matter what GPCR we're eventually going to signal, we want these two hormones to end up increasing our blood sugar, right? So glucagon has only one receptor, the glucagon receptor. This glucagon receptor is coupled to a G protein. That G protein is G protein S alpha. It activates the enzyme adenylate cyclase. We saw this in the animation, right? Adenylate cyclase creates the second messenger, cyclic AMP, right? We saw that in the video. Epinephrine, if it binds to the beta-2 androgenic receptor, that beta-2 androgenic receptor is also bound to G protein S alpha. So these two, if you are bound to the same thing, you are in a shared pathway, right? Even though we have two different signaling molecules, it's the same thing, because if we have G protein S alpha, that's going to activate adenylate cyclase. Adenylate cyclase is going to make cyclic AMP. We're going to go through the same thing. But remember how I said I was going to share, I was going to spoil it earlier? I said epinephrine can bind to a second receptor. It can bind to the alpha-1 androgenic receptor. The alpha-1 androgenic receptor is bound to GQ alpha, not the same G protein. This G protein activates something different, phospholipase C. Do you remember we talked about phospholipase C in the last section? Yep, phospholipase C cleaves what? Well, you're going to make IP3, PIP2. You're going to cleave PIP2 and you're going to make DAG and IP3, right? So eventually, these molecules and second messengers are going to end up doing the same thing, eventually increasing your blood sugar levels, but they're going to go through a different pathway, right? So this is going to give you a shared pathway, right? So glucagon, yeah, shared. So this is going to be, if I do this, right? These are parallel, P-A-R-A-L-L, parallel pathways, right? Because we have the same stimulatory molecule, the same ligand, epinephrine, but it activates two different GPCRs. They're different because they're bound to two different G protein molecules. And because the G proteins are different, they activate different downstream signaling molecules. But eventually, what's the whole goal? Release glucose. Okay, so shared pathways and parallel pathways. Be able to tell the difference between the two. All right, so now let's follow one GPCR through its entire signaling pathway, right? So the first step is we have some kind of ligand activating. So our ligands are, for example, epinephrine, glucagon, right? It binds to the G protein coupled receptor. What happens to the G protein coupled receptor? We have a conformational change that gets propagated from the extracellular side to the cytosolic side. The cytosolic side will change. Normally, when unbound, all this stuff is together. But when that ligand binds, you're going to have disassociation of this G protein molecule. And that disassociation is because you exchange GDP for GTP. So normally, in the inactive state, you have GDP bound. When the receptor binds, you induce that conformational change, you switch out GDP for GTP. Now that we have GTP bound, things disassociate, right? Once those different subunits of the G protein disassociate from one another, now they can go on and activate all kinds of downstream signaling events. We're going to talk about what those are. Okay. So each one of the different G proteins, each one of the different subunits can activate different things, right? So do you remember how we said that there were two of them that were GS alpha? What does GS alpha activate? Adenylate cyclase. Do you need to know that? Yes, yes. Okay. How about my other one? What was the other one that epinephrine activated? GQ alpha. GQ alpha stimulates phospholipases. Why would I want to stimulate a phospholipase? Well, that's the pathway. Yeah, there's a yes, that's the pathway that happens. But we want to cleave. We want to make second messengers instead of making cyclic AMP, right? This is going to go on and make cyclic AMP. We want to go on and make PAG, IP3, right? So it depends on what other downstream signaling molecules you want to activate. And these are second messengers, right? Second messengers. This one, those, right? And they have different effects. That's what we learned in the first section. And there are others, and they do other things. But these are kind of the big ones that we're going to talk about. Okay. So here's our step-by-step just in text of what we just said, right? Our ligand binds, induces conformational changes in the receptor. The receptor, because it's in the bound state, now exchanges the GTP, well, the GDP for GTP, right? Now that you've exchanged it and you are bound to GTP, now you can upregulate all your downstream effectors and then activate your second messengers. But what's really important is that we also talk about some way that we terminate the signal. Okay, that's absolutely something that is insanely important. Otherwise, this signaling will go on forever with just one ligand, and that is not what you want, right? That's what's happening with my birthmark, actually, right? It's actually constantly growing. Like I have to go every so often and get laser treatments on it because it continues to grow. Oh, and by the way, FYI, if you ever get a tattoo, make sure you really love it because it is painful to get it removed because it's the same process. It's the same laser treatment. So if you ever want to know how painful it is, I'll tell you. It hurts, especially on the face. What's that? They basically kill the cells that are holding on to the ink, to the dye. Yeah. Probably every, let's see, when the last time I did it, it's probably been like a year and a half just because it's been coronavirus. But I like to go like every year. That's what I prefer to do, but I don't always. Like when I was a kid, I had, oh my gosh, like 17 or 18 different laser treatments, like in a row, like back to back. It's not fun, but it's lightened quite a bit. So I'm really happy that I did it. But the point is that the receptor is always on to stimulate blood vessel formation. So that's what happens when you have a mutation that prevents the signal from being turned off. If it's never turned off, it's just going to continue to grow, or it's just going to continue to tell that you taste sweet and you would always taste sweet, right? That would kind of suck. No change. All right. So depending on what ligand you get will bind to a specific receptor. That specific receptor is bound to varying G proteins, right? That's what we just said. Each of those G proteins activates a different downstream effector molecule, which activates different secondary messengers. And that's what we see in every single one of these. And this is just 3 examples, right? There are many, many, many. Okay, so let's kind of go back to this is back to our big scheme, right? So let's kind of, before we do this, okay, so if we have a ligand, our ligand activates our G protein coupled receptor, right? Okay, our G protein coupled receptor is bound to a G protein. What happens to the G protein before it dissociates? You exchange GDP for GTP. Now that you have that, you can have dissociation of G protein subunits. Okay. When they disassociated, what enzyme did we recruit? Adenylate cyclase. This is a really good study method, right? Like, can you write out the pathway of what happens? Okay, so we recruit adenylate cyclase. What does adenylate cyclase do? It doesn't recruit cyclic AMP, it converts, it's an enzyme. Takes ATP and makes it cyclic AMP. So from 3 phosphates to 1 phosphate. Okay, now what? Now what does this cyclic AMP do? Remember from section 1, we talked about it, what it does in section 1. So if you have high levels of cyclic AMP, what happens? We activate pKa. We activate pKa. What is pKa? So this is the problem when you get into all these abbreviations, right? Protein kinase A. So it is a kinase. A kinase can go in and do what? Phosphorylate. P-H-O-S-P. Is that right? No, I think I did it twice. Whatever. Y'all know what I mean. That's fine. Whatever. There's too many. Whatever. There's too many letters in there. All right. So what is it going to phosphorylate? Phosphorylase. Kinase. I'm having trouble today. Okay. K-I-N-A. It's a good thing you don't have to spell to be in science. So we're going to phosphorylate phosphorylase kinase. Do you remember this? Sort of, kind of. Everybody's eyes are bugging out at this point. So what does phosphorylase kinase do when it becomes active? It is going to, we're just going to do this. We're going to do a couple of arrows, and then what happens? What's our like end product? Glucose. We want to get glucose from glycogen. So G-L-Y-C-O-G, glycogen to glucose 1-phosphate. And that goes to the blood. You should be able to do that. Practice that over break.
Right, the test is before.
Oh, the test is the weekend. I meant the weekend. I meant the weekend. We could do that. No, I meant over the weekend. This is just wishful thinking on my part too. No, I need the break to grade job papers. Okay. So let's come all the way back here. So now, protein kinase A. Now we know where protein kinase A fits into our pathway, right? Because you just wrote the pathway. You wrote what I wrote, right? So protein kinase A is a downstream signaling protein. Why do we call it a downstream signaling protein? Because it is below the second messenger, cyclic AMP. Okay, let's go back up. All right. So it's 3 distinct metabolic responses that happen when you get high levels of cyclic AMP, right? So what we want to do is we want to increase our blood sugar, right? So what we've talked about so far is this second one, phosphorylation and activation of enzymes involved in glycogen degradation to produce glucose. That's the one we have already talked about. But a lot of times what you're going to see is you have multiple things that happen in the cell that lead to basically the same overall physiological effect happening. So at the same time that you want to activate enzymes that produce glucose from glycogen, so we're going to say activate glycogen degradation. That's kind of our big key there. At the same time, we want to inhibit glycogen synthesis, right? Because if we are synthesizing glycogen, what are you doing? You're dropping your blood sugar. That's not what we want to do. We want to increase our blood sugar. So a lot of times when you have 2 pathways that do the opposite thing, it's like a seesaw. You don't just increase one. At the same time you increase one, you decrease the other. So if we're going to increase degradation, we want to decrease synthesis, and vice versa. When you would eat a meal, the same thing would happen. You would inhibit glycogen degradation and you would activate glycogen synthesis. So that's the seesaw. The other thing that you're going to do is you're going to also activate enzymes that are involved in gluconeogenesis. What is gluconeogenesis? Formation of glucose from other metabolic sources. So all of these 3 things lead to what? All 3 lead to what? Not an increase in BS, but an increase in blood sugar. That's all y'all are going to think about. I know it. Okay. Aren't y'all glad we drew out the pathway for that? Like, sure, no, not really.
No, I am.
Oh yeah? Okay, good. All right, so this is kind of a nicer drawing, but I feel like it's really important that you can kind of do it on your own without having to follow a picture. But it's nice to have a picture to go back to. So here's our same pathway, right? We have epinephrine. Epinephrine binds to our G protein coupled receptor. Our G protein exchanges GDP, that was in the bound inactive state, for GTP. Now that it's active and bound to GTP, it activates adenylate cyclase. Adenylate cyclase is an enzyme that converts ATP to cyclic AMP. Cyclic AMP is the second messenger that activates protein kinase A, right? Protein kinase A will then go on and basically affect glycogen metabolism, right? In 3 different ways that we just talked about. But the end result is the same. We have net glucose export, right? Okay. So at the same time, we also have to think about shutting off this mechanism. We don't want it going on forever. So cyclic AMP, when it activates protein kinase A, it's done its job. So basically we need to turn it off. So protein kinase A will go on and affect glycogen storage and glycogen degradation, but it will also allow cyclic AMP phosphodiesterase to work. So cyclic AMP phosphodiesterase will convert cyclic AMP to AMP, which is not a second messenger. So this is a way for us to stop the signal. Okay. All right. Now, this was which kind of receptor? The beta-2 androgenic receptor, right? But we said there's a different pathway if you have an alpha-1. So let's look at the alpha-1 pathway. In our alpha-1 pathway, we still have a G protein, but this G protein is G protein Q alpha. G protein Q alpha, when it's bound to GTP in the active form, it's going to stimulate PLC. What is PLC? Phospholipase C. Phospholipase C will cleave PIP2. Remember, PIP2 is partially in the membrane. Well, it's really embedded in the membrane, right? And when you cleave off the two major portions of it, what are you going to get? You're going to get a diacyl glycerol, and you're going to get IP3, basically a sugar, right? So these are what? Second messengers. So they can do two different things. IP3 mediates calcium release. Well, guess what? Glycogen degradation needs calcium in order to go. So that's going to turn on glycogen degradation because you have enough calcium present in the cell. DAG is going to activate PKC, protein kinase C. What does protein kinase C do? Turns off glycogen synthesis, and it actually turns on glycogen degradation, right? So really there should be another arrow that does this number. But that's okay, we'll forgive the book. They got a lot going on. But the whole point is, no matter if you're activating the beta-2 or the alpha-1, what's our end result? Increase in BS. Increase in our blood sugar due to glucose export from the cell. I'm never going to live that one down, am I? It's all right. All right. Yeah, sure. Good. Okay. So we need to talk about a couple of control mechanisms that occur for GPCRs. So GPCRs are controlled by two things. Those two things are GEFs and GAPs. A GEF is a guanine nucleotide exchange factor. So what do you think it does? It exchanges nucleotides that have guanine, right? So this promotes our exchange from GDP, excuse me, to GTP. So we are going to activate our signaling. This disassociates our G protein molecule, right, and allows them to do whatever signaling they're going to do, right? So we're right here. Now, you have GAPs. GAPs are GTPase activating proteins. What is a GTPase? It breaks down GTP into GDP. So it stimulates intrinsic GTP activity. So you go from GTP to GDP. So here's an example of a gap. So if the gap comes into contact with a G protein that has GTP associated with it, it's going to cleave that GTP into a GDP. Now, the G protein can reassociate with our receptor, and now it is in the what? This is now inactive, right? So this is active. So if we have GTPase activity, we are going to inhibit our signal transduction because we're going to return this receptor back to the inactive state. And that's really important that we have a way to reset the receptor. Okay? All right. So once this G protein, all the subunits are reassociated, right? We want to be able to turn it off. We want to be able to desensitize it, right? So our G protein-coupled receptor kinases will go in and phosphorylate the cytoplasmic tail of our receptor. And it'll phosphorylate serine and threonine residues and V residues, threonine residues, right? Okay, so once the receptor goes off, you want to be able to put it into a state where it can't continue to signal. So the way that we do that is by phosphorylation. So remember in the video, right, that there was one of these that came in and phosphorylated the cytoplasmic tail, and then what happened to the molecule? It got endocytosed, right? So that's exactly what's going to happen. It's going to provide a docking site for a protein called beta-arrestin. Beta-arrestin prevents everything from getting back together into that ready to go primed complex, right? We not only want to shut off the signal, but we also want to prevent it from reassociating and being able to signal again and again and again and again. So if you phosphorylate the tail of that receptor, it now interacts with beta-arrestin. And think about arrestin, right? If you arrest something, it can't do anything anymore. And that's the whole point, right? So here's our, let's see where we are. Here's our G protein coupled receptor, right? Everything has basically disassociated. It's doing its job. So what will happen is you'll have protein come in and phosphorylate our cytoplasmic tail. Now beta-arrestin associates with that cytoplasmic tail. That induces endocytosis. So this is kind of a terrible diagram, right? But you have that G protein coupled receptor, right? And it's got its phosphorylated groups. And basically it blebs inward, and the protein and the membrane circles around, and you create basically a pocket, and it traps the receptor inside the pocket, right? So now it's in this endocytic vesicle. And basically what happens is, depending on the cell type, there are two things that could happen. You could actually have proteolytic enzymes that would come in and actually completely destroy it. It would chew it up and kill it, right? So then in order to get more receptors, what would have to happen? You'd have to start from scratch, right? You'd have to start at the DNA. You'd have to make some RNA. You'd have to express the protein from that, right? So it could be dependent on transcription at the DNA level, or you could just have them sequestered for a short amount of time, and then they're recycled back to the plasma membrane. So it depends on what cell type and kind of what needs to happen. Okay, so that is G protein coupled receptors. Good Lord. Thought we were going to get more 3-2, but that's okay. It's okay. It's okay. All right, so how about we watch the video one more time, because that's going to be perfect, and I think that's going to be a really great solidify it in your brains sort of deal. Y'all okay with that? Okay. Sure.
Reception of vision, smell, and taste are all mediated in part by G protein coupled receptors, or GPCRs. GPCRs should evolutionarily conserve structure consisting of a central core made up of 7 transmembrane alpha helices. In the cytosol, GPCRs interact with a membrane-bound protein complex called a heterotrimeric G protein, which consists of G alpha, G beta, and G gamma subunits. The G alpha subunit is a GTPase and can hydrolyze GTP to GDP. When the GPCR is not bound to the ligand, it is bound to the inactive GTP-bound G alpha beta gamma complex. In most GPCRs, an extracellular signal induces a conformational change in the GPCR protein. The conformational changes in the activated GPCR stimulate the exchange of GDP for GTP in the G alpha subunit. GTP-bound G alpha dissociates from the heterotrimeric complex. Both G alpha and G beta gamma can then initiate distinct downstream signaling pathways. In some cells, different GPCR-mediated signals are integrated in response to different extracellular stimuli. Liver cells, for example, must respond to the hormones glucagon and epinephrine in order to regulate glucose levels in the bloodstream. Both glucagon and epinephrine activate a specific G alpha called GS alpha after binding to the glucagon receptor or the beta-2-adrenergic receptor. After exchanging GDP for GTP, GS alpha undergoes a conformational change in a specific region of the protein called the switch 2 helix, so that it loses affinity for G beta gamma and interacts with an enzyme called adenylate cyclase. This interaction stimulates adenylate cyclase, resulting in the production of the second messenger molecule, cyclic AMP. At low levels of cyclic AMP, protein kinase A is bound to a regulatory subunit within an inactive tetrameric complex. As cyclic AMP levels rise, cyclic AMP molecules bind to the regulatory subunits, inducing a conformational change that releases the active protein kinase A monomers. Protein kinase A activation triggers several downstream pathways that all lead to glucose export into the bloodstream. One of the substrates for protein kinase A is the cytoplasmic tail of the Beta-Androgenic Receptor. The phosphorylation of the GPCR by protein kinase A and other regulatory kinases provides a docking site for beta-arrestin, a protein that results in downregulation of GPCR signaling and endocytosis of the receptor. The over 600 GPCRs in our body recognize a wide variety of signals, such as light, compounds we can taste and smell, and hormones, to enable our cells to perceive and react to environmental cues.
So second time, do you get more? Yeah, right? Okay. All right, so have a good weekend. Study, 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 study. More than likely not. We probably won't get through everything, but we will have an exam. We'll cover what we cover on Monday. We will have an exam on Wednesday. Y'all have a very good weekend. That's a different kind of question than at a parent's house. Is that chocolate? I don't want to know. I want to know.
I didn't see that first. I just see, no.
All right, let's see. Anybody have anything on the chat? Okay,
It's not
must be good. All right, we're gonna end.
Gotcha.